Hello, everyone. Welcome to today's live broadcast, Beyond the Hype, the evidence behind the role of genetics in drug response. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational seminar is presented by LabRoots, the leading scientific, social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. Now, if you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or you can submit your problem through the green Q&A button on the lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. I would now like to introduce today's speaker, Dr. Matt Nelson. Matt Nelson is the head of genetics at GSK, working in Philadelphia, Pennsylvania, leading a group of scientists to bring genetic evidence into both drug discovery and drug development decisions. Research activities of interest include investigating the role of the growing body genome-wide association studies to inform drug target selection and validation, improving pharmacogenetics experiment design and developing methods and strategies for drawing inferences from both small and large-scale genetic association studies. Other recent activities include analysis of deep resequenced data for target validation, development of the PopRes genetic resource for pharmacogenetics research, and co-chair of the Scientific Management Committee of the Cross-Industry International Serious Adverse Event Consortium, ISAEC. Matt's path to research in the pharmaceutical industry began at the University of Michigan, where he received degrees in statistics and human genetics with an emphasis on novel combinatorial strategies for identifying gene-by-gene interaction. Upon graduation, he worked with the startup biopharmaceutical company Esperion Therapeutics for 3 years. At Esperion, his activities included cheminformatics, preclinical and clinical study design and analysis, development of online analysis and reporting tools, and leading the information systems group. Matt joined Sequenom, a San Diego-based biotechnology company, in early 2002 as director of biostatistics, where he was involved in some of the earliest attempts at genome-wide association studies. Matt has been with GSK since 2005, with prior roles as researcher and head of statistical genetics. I would now like to turn the presentation over to Dr. Matt Nelson. Dr. Nelson?
Thank you, Christy. It's a pleasure to be a part of this virtual event around precision medicine, talking about the current evidence for the role of genetics in drug response, both in terms of safety and the efficacy. So I'm going to start out this presentation with a little bit of background on genetics research. This is going to be very superficial. I'm not going to go very deep given the time constraints that we have and the other material that I hope to be able to cover. I'll give you a little bit of a glimpse in terms of what were some of the early hints that genes could play a really important role in how we respond to some drugs, and the series of research activities that has led to, both from the perspective of understanding drug safety as well as understanding drug efficacy. And I'll finish that off by discussing the implications that this research has had for our own research activities within GSK, a drug discovery and development company, and how we incorporate genetics into our routine drug development activities. Great. So first, a little bit of genetics background. So genetics is the study of heredity. It's the study of how the genetic characteristics that we inherit from our mother and our father influence the way that we respond to the world around us. from a phenotypic perspective. So going back to your high school and college genetics course, of course you know that there are 23 chromosome pairs. We inherit one copy from our mother, one copy from our father. Every single cell in our body, every live cell in our body, has a full complement of these pairs. There are 6 billion bases in our genetic material within each cell. It's truly remarkable. If you were to take the genetic material out of one cell and unwound it, it would be roughly a meter long. But of course, that gets packed into a submicroscopic level inside of each of our cells. Within that DNA, it codes for about 20,000 so-called genes. These are stretches of DNA that code for the proteins that are so integral in our biology and in who we are. Now, although if you compared any two individuals, we roughly have the same DNA, there are a lot of differences from person to person across our DNA, and that's what gives rise to genetic variation and heritability of our individual traits. There are roughly 10 million relatively common single nucleotide changes. We call those single nucleotide polymorphisms across our DNA. And there are many, many more very rare variants. There are other kinds of variants that we inherit. One type is called an indel or an insertion deletion. These are relatively small, 1 to, say, 15 base deletions of those letters that make up our DNA. There are also more complex changes as well. They can be very large deletions of anywhere from 100 bases to hundreds of thousands or even millions of bases, or rearrangements of those DNA. All of those give rise to differences in our individual characteristics. Just to remind you, this is the canonical cartoon of a gene. You have a relatively long chromosome, a stretch of DNA. Within that chromosome, you can identify regions of that DNA that code portions of proteins. So in this cartoon, we have 4 protein coding portions that contribute to gene X. We call those protein coding portions that are shown here. I can get my annotator out. They're shown here. Those are called exons. The segments of DNA that separate the exons are called introns. And so if you zoomed in on any section of that gene, you could see it doesn't actually look like letters, but we use the letters to represent the chemical building blocks that make up our DNA. And again, so this represents a single nucleotide polymorphism, a change that may occur at any one of these bases that gives rise to the genetic differences amongst us. And those genetic differences we can actually measure, and there are a wide variety of different laboratory techniques, from techniques that use silicon-based chips to techniques that actually go in and sequence each individual base, where we can measure those underlying genetic characteristics in each person in our sample. And we can take those underlying genetic characteristics and we can relate them to differences among individuals. So here I'm showing an example that I'll come back to in a minute, but on the y-axis here of this plot, we have the dose of warfarin that patients have received. And across the x-axis here, we have different genotypes. And again, these genotypes are referenced as 2 bases, because the one base you get from your mother and the one from your father. So we have some individuals that are TT, some individuals that are TC, and some individuals that are CC. And in this particular instance, we find that there's a relatively large effect of that genotype on the dose of warfarin that a person ends up on once they have been titrated to the proper dose. This is one of the first discoveries that we had early on in pharmacogenetics, and that's the vitamin K receptor for warfarin. And we found that differences at this site had a relatively big impact on the ultimate dose that a person would settle on. So again, we can measure genetic variation in a large number of ways. Very modern panels that are used today can measure anywhere from 300,000 to 1 million of these single nucleotide polymorphisms. They can measure thousands of insertion deletions. And we can use those measures to carry out studies like I was showing in the previous slide all the way across the genome. And we refer to those studies as genome-wide association studies. And so in each of those studies, we can look at every single one of those variants and ask, is that variant related to the disease or to the characteristic that we're interested in studying? You don't have to rely on panels. We actually have the capability now to sequence either the entire exome, which is a word that just refers to the coding portions of our genome, or even the entire genome in one fell swoop, revealing tens of thousands of genetic variants at the individual level that we can then study. So our capability to delve into, to measure our genetic material, and then to relate those differences in our genetic material to our various characteristics has just been transformed and revolutionized over the past 10 years as the technologies for doing this have developed. What I'm showing here is the discoveries that have been made using that type of genetic technology. And if you can see the x-axis here, the scale that, let me turn my pointer here, so across the x-axis, oops, wrong way, there we are. Across the x-axis we have dates, and we're essentially showing the accumulation of these discoveries of how our genetic variants influence our traits over the past roughly 12 years of discovery. On the y-axis is the number of discoveries that have been reported. And these are coming from a catalog that I'll show you in just a moment. But you can see that back in 2005, there were very, very few. So here, in 2005, there were just a very small handful, maybe 10 to 20 of these genetic variants that were influencing human traits that we had actually been able to discover and validate. You can see that starting in roughly 2007, the pace of those discoveries began to pick up quite dramatically. And this coincides with the development of the technologies to be able to measure human genetic variation at such a large scale and conducting these genome-wide association studies. The pace continued to go up quite rapidly. That increased pace was partly influenced by further improvements in technology and what we could measure, but the main advancement in those discoveries was around the size of the studies that we could carry out. As we carried out larger and larger studies, more and more cases, more and more controls to identify how our genetics is influencing our traits, we found more and more under the hood, more and more of those individual genetic variants that are having an impact on so many different characteristics. You can actually go out and query those discoveries yourself. There is a site on the internet called the Jiwa's Catalog. This is hosted by the European Bioinformatics Institute, EBI, after it was transferred there from the United States NHGRI, the National Human Genome Research Institute. And you can type in any gene that you're interested in or any disease that you're interested in, and you could identify all of the different genetic variants that have been discovered to be involved in that disease. So here's a little example of the kind of query results you could get. So I've inserted here a drug response phenotype, liver injury, and by typing that in, I found that there were several publications for several different drugs where they had investigated the genetic impact on the liver injury phenotype. And you can see that for each of these, down here in this last column, indicates the number of discoveries underlying each of those. If you were to do this, you could click on the little plus there on the far right, and you can explore those in greater detail to find where in the genome those discoveries were found. Okay, so that's a little bit of background on genetics. So let me get back to the topic at hand, and that's pharmacogenetics. Pharmacogenetics is simply a term that refers to the genetics of drug response. Drug response can be described in terms of drug exposure, how much drug we get in our system when we take a prescribed dose. It can refer to drug safety, adverse reactions such as liver injury. It can also refer to drug efficacy. How well do we respond to a drug when we receive it? Genetics has been shown to have some influence in all of those, and we collectively refer to the study of these three things as pharmacogenetics. Pharmacogenetics is not a recent discovery or phenomenon. Actually, the first publication for pharmacogenetics was published by the famed geneticist R.M. Matulsky in 1957, where he described protein differences in G6PD and the impact that had on anemia and antimalarial response. So pharmacogenetics is not at all a new science. I showed you earlier one of the early efficacy pharmacogenetics discoveries, and that is the influence of this genetic variant in the vitamin K receptor on warfarin response, and essentially the dose that a person eventually is prescribed when you titrate to a particular response, coagulation response in the blood, is strongly influenced by variants in the vitamin K receptor. So with this background, and so this vitamin K receptor association was discovered in 2005. There were a lot of expectations that genetics would be applied to drug response, and that we would find right and left these big genetic factors that would help us to identify just which patients a drug will be safe and effective in. And this was particularly of interest because we've seen over the years that drug discovery and drug development is very hard. This pie chart on the right shows the reasons why drugs fail in phase 3. And we know, of course, that roughly 50% of all drugs in phase 3 do fail phase 3 clinical trials and don't progress to registration. And the primary explanation for these failures are due to lack of efficacy, 64%, and insufficient safety, 27%. So an ability to use genetics to better differentiate the best patients to receive a drug could potentially have a very big impact on drug discovery and development, and ultimately on ensuring that patients are getting the right treatments. Also around that time, in 2001, this is actually abacavir, is a drug that GSK developed for treatment of HIV. And we had found during the drug development and clinical trials that up to 5% of patients that received abacavir would experience a hypersensitivity reaction. And that hypersensitivity reaction could be quite severe. And if a patient continued to receive the drug or was rechallenged with the drug, that reaction could potentially be life-threatening. So there was a whole series of mitigation strategies that were put into place to be able to identify very early signs of these reactions and to withdraw patients from abacavir if they were seeing that. Well, we started applying our genetics tools, as crude and rudimentary as they were back in 2000, to see if we couldn't identify genetic factors that might differentiate the patients, the 5% of patients that received the drug and demonstrate some sort of hypersensitivity reaction from the 95% that received the drug and never experienced any type of negative adverse event. And lo and behold, what we found was that the portion of our genome, the HLA region, that encodes the proteins that are involved in our adaptive immune response, a particular version of the HLA-B gene called 5701, was actually very strongly associated with these hypersensitivity reactions. And that's what's shown here in this table. In the first publication describing this with just 84 cases and 113 controls, we see that those that had HLA-B57 were much more likely to experience the adverse event than those that did not carry HLA-B5701. So GSK set out to definitively demonstrate the role of B5701 in hypersensitivity reaction through a pair of clinical trials that were carried out. One of these clinical trials was called PREDICT-1. In a PREDICT-1 study design, we took patients that had HIV that had never received abacavir before, here referred to as ABC or abacavir-naive subjects, and they were either randomized to the standard of care, which is to prescribe abacavir and to follow the patients very carefully, withdrawing them if they show any symptoms of hypersensitivity reaction. And in the other arm, in equal numbers, we carried out a pharmacogenetic screen, and we removed patients from the clinical trial that carried the B5701 allele, that were positive for that allele, and we only gave the drug to patients that did not carry that risk factor. The results of the PREDICT-1 trial were published in 2008, and the results were overwhelmingly positive. What we found was, in particular, when we looked at patients that had a skin patch test that was a objective confirmation that they were experiencing a hypersensitivity to abacavir, we found that in the arm in which no patients carrying B5701 received abacavir, there were no events of hypersensitivity reaction. In the other arm, where they were randomized to the standard of care, we found that only patients carrying B5701 experienced and immunologically a skin patch test confirmed hypersensitivity reaction. So this test we found was 100% predictive of being able to take abacavir safely. So again, this came relatively early on in our understanding of genetics in drug response. And so there was tremendous optimism and enthusiasm that we would be able to explain away most negative effects to drugs through application of genetics. And in the instance of abacavir, this test made it into the drug label, and since has had an important role in the use of abacavir in treating patients with HIV. So what have we learned since then? Has genetics been this treasure trove of information about how patients are going to respond adversely to drugs, and is it having a big impact in the clinic? Well, so this table here comes from a publication that came out, I was the first author on in 2009, that summarized what was the state of the field in 2007, roughly around the time that this abacavir story was coming out. And there were very, very few such discoveries at that time, and actually looking across this list, there are a few of these discoveries that probably don't really hold up. Well, we within GSK and really across the industry and in academia as well, we're quite enthusiastic about this as a strategy for improving overall safety of drugs. So we saw this as essentially a call to action. In 2007, we knew that genetics could play an important role in serious adverse events, such as abacavir hypersensitivity. We knew that collecting cases and studying genetics, especially at that time, was expensive and challenging. We didn't know what the best methodologies for studying serious adverse events with genetics would be, but we knew that it was in everyone's common interest to study this as thoroughly and effectively as possible. So there were 10 different pharmaceutical companies and several public and academic groups that came together under the International Serious Adverse Event Consortium with the goal of understanding the mission to identify DNA variants useful in understanding the risk of drug-induced serious adverse events. and ultimately in being able to predict those patients that would likely experience a serious adverse event. A big emphasis of this consortium was to pool both the commercial and academic resources to be able to identify the cases of the serious and generally very rare adverse events so that we can study them sufficiently and develop the methodology around that research. The initial focus and primary focus of the SAC, shown here, was primarily around liver injury, skin injury, including hypersensitivity reactions, eosinophilia and systemic symptoms, renal injury, torsades de Pointes, as well as several inflammatory bowel disease therapy-related SAEs that are observed. We also were able to opportunistically investigate a number of other conditions, including osteonecrosis of the jaw, excessive weight gain due to atypical antipsychotics, and even clozapine-induced agranulocytosis. As I said, an important part of this cross-academic commercial consortium was just bringing all of the researchers together necessary to identify large enough numbers of serious adverse event cases to be able to study them from a genetic perspective. And what's important to note is that, for example, liver injury, there are literally hundreds of drugs that are reported to potentially have some impact on liver safety, on liver. And so to be able to get enough cases of drug-specific events, it really takes this type of international consortium to carry it out. And this table here summarizes the success that we achieved in doing that. We had almost over 2,500 liver injury cases. We had nearly 1,500 serious skin reaction cases, and you can see the numbers amongst these others. So all in all, the Serious Adverse Event Consortium was instrumental in bringing together over 7,000, nearly 8,000 cases of these various serious adverse events. I'm going to show you now a couple of discoveries that kind of help illustrate how we do this research, as well as the kinds of results for this research. What I'm showing you on this slide was our very first publication in 2009, really our first success that came from this consortium. And this is the result of a GWAS, or a genome-wide association study, for flucloxacillin liver injury. Flucloxacillin is an antibiotic. It has an incidence of liver injury, and sometimes very serious liver injury, of approximately 1 in 5,000 cases. And we were able to, very early in the consortium's life, bring together over 50 cases of this serious adverse event. And we, with those 50 cases and a large number of controls, we measured them genome-wide. So we had roughly 1 million genetic variants that we were able to measure in these patients. And for each one of those 1 million variants, we carried out an analysis of whether that variant was associated with the risk of liver injury due to flucloxacillin. So across the x-axis in this figure, which is often referred to as a Manhattan plot, we have the location of each of those 1 million variants that we tested from chromosome 1 up through chromosome X. And the height of each of these points is the p-value, or the statistical significance of the relationship between each variant and the risk of liver injury due to flucloxacillin. Now, if you're used to running kind of your traditional statistical tests, one hypothesis at a time, you may recall that the approach is to pick some p-value threshold that would lead you to make a good choice as to whether to reject the null hypothesis. And traditionally, for a single test, that value would be on the order of about 0.05 or maybe 0.01, or if you're going to be really stringent, 0.001. Well, that's fine if you're only conducting one test, but if you're conducting 1 million tests, you have to be much more careful about how you guard yourself from making a type 1 error, and that is rejecting the null hypothesis when in fact it is true. And so you have to use much more stringent statistical criteria. So for a traditional genome-wide association study, we would be using thresholds on the order of about 10 to the negative 8, so about right here, for drawing our conclusions. It's roughly at 10 to the negative 8, so a p-value that has a 0.0, well, 7 zeros and followed by a 1 after it. And you can clearly see that there is only one region of the genome that exceeds that threshold, and it exceeds that threshold by far. We have p-values that are down around 10 to the negative 35. That peak that comes up here, and there is only one peak from this association, falls under the HLA region of the genome. This is the same region that we saw previously associated with the back-of-your hypersensitivity reaction, and lo and behold, it actually turns out to be the exact same genetic variant, that is HLA B5701, underlying flucloxacillin liver injury. So that was our first discovery in that area. Our next discovery was also in liver injury, and that was for coamoxiclav, or amoxicillin clavulanic acid, which here in the United States is referred to as Augmentin. For this Augmentin GWAS, again, so you're seeing a very similar Manhattan plot for this, and you're also seeing a relatively similar pattern, and that is that we don't have very many p-values that get small or reach that genome-wide significant threshold of 10 to the negative 8, except in the HLA region. What was interesting about the Augmentin liver injury study was that when we zoomed in on that region of the HLA, what we actually found is there were two independent peaks of association. One peak over here on the left is associated with class 1 HLA molecules, and this region over here on our right associated with class 2 HLA molecules. And so again, we find that HLA is playing a pretty important role. Well, we've carried out really hundreds of such analyses looking at different types of serious adverse events with and the risk provided by different drugs. And I won't bore you with each of those, but what I will do is summarize for you essentially where the field of genetic influence on adverse events is today. So I showed you a few slides back where we were in 2007. Well, this is a similar slide showing where we are today. On the left are the discoveries where they're grouped into different types of adverse events, liver injury, skin injury, some of these other hypersensitivity reactions, and then just a catch-all for everything else. These are the discoveries that were made outside of the Serious Adverse Event Consortium. So some of these from GSK, some of these from other pharmaceutical companies or academic researchers. On the right are the discoveries that have resulted from the consolidated efforts of the Serious Adverse Event Consortium. Let me just focus in on a couple of key messages from this slide. One of the key messages is the kinds of associations that we're observing. So the associations shown in red are those that were localized to the HLA locus. So these are immune response related adverse events. The ones in green correspond to genes that are involved in drug metabolism, the absorption, distribution, metabolism, and excretion of a drug. So one of the key patterns that you can see based on these relatively well-validated discoveries to date is that all of these associations fall into one of those two classes, either HLA or ADME. The other point to make is that the Serious Adverse Event Consortium has been quite productive in terms of the discovery of a very large fraction of the total discoveries that we've had to date in this area. One of the questions you might ask is, well, if you're familiar with some of the limitations of genome-wide association studies, well, genome-wide association studies are generally limited to investigating relatively common genetic variants. But the genetic variants underlying, or well, most of these serious adverse events are quite rare. So is it possible that there are also very rare genetic variants that might be underlying these rare adverse events? So the Serious Adverse Event Consortium has looked at 3 different adverse events in some detail where we didn't just use genetic markers across the genome, but where we actually went down and sequenced at the variant level the participants in these studies. And what we found, so the 3 studies we carried out were augmentin liver injury, the clozapine-induced agranulocytosis, and Torsades de Pointes. And really the bottom line there is so far we have not found that rare genetic variants are primarily responsible for those rare adverse events, but rather that we're finding relatively common variants that are involved. Another lesson that we've learned as we've elucidated the growing number of genetic risk factors for some of these serious adverse events is that, again, the genetic risk factors tend to be relatively common, whereas the adverse events tend to be relatively rare. So I'm circling here the frequency of the variants that we discovered in these populations. They range from anywhere from 1%, sometimes a little bit smaller than 1%, as low as 0.002%, or 0.2% up to about 30%. While these variants can have relatively large effects, they don't tend to be very predictive. That is that the vast majority of patients who carry those variants can take the drug and will never experience that adverse event. So whereas for the example I showed before, abacavir, roughly 50% of the patients that carry B5701, if they received abacavir, would end up having that adverse drug reaction. For the other kinds of discoveries that we found so far to date, most of them look a bit like this. That is that because the adverse events are so rare, the vast majority of the patients that carry these genetic risk factors, even taking the drug, will never develop them. It's even more challenging when you look at the negative predictive value. Again, we found that essentially the negative predictive value, or 1 minus negative predictive value for abacavir, was 0. That is, that those that didn't carry the risk allele would never develop the adverse drug reaction. For the other drugs and risk factors that we've identified, the genetic marker doesn't do that good of a job of differentiating those who would go on to develop the adverse reaction versus those that won't. On that topic, I want to introduce you to a very valuable resource. This is the Clinical Pharmacogenetics Implementation Consortium. I have the website for it down here in the lower left-hand corner. The goal of CPIC is to essentially look at the evidence that's out there for each of the drugs and the genetic variants that are associated with those adverse events and assess what impact those might have clinically. And if you go to that website, you can find 33 published guidelines that provide a summary of the science underlying each of those associations, as well as clinical recommendations for that. And that really is, I think, the single best resource that I could point you to if you want to have a better understanding of whether there are genetic factors that might have an impact in a particular drug that you are interested in prescribing and potentially considering a genetic test for. So what have we learned so far? Let me go through this quite quickly. The mechanisms underlying the genetics of serious adverse events are largely limited to HLA and ADME, these drug metabolizing genes. Common variants with a significant impact on the risk of serious adverse events can be identified with relatively small numbers of cases, as few or even smaller than 50, using these genome-wide approaches. What we find is that most of the HLA genetic risk factors are fairly drug-specific, although we do see some commonalities like the B5701 that we saw for both abacavir and flucloxacillin. The HLA risk factors appear to be fairly serious adverse event specific for a given drug. And as I mentioned in the previous slide, common genetic risk factors alone don't accurately predict patient risk for rare SAEs. And as such, there aren't many instances where prospective typing is going to be justified when making a decision about which drug a patient should receive. And the serious adverse events for many drugs, really more than 50%, don't appear to have a major genetic risk factor. So we don't expect genetics to explain much of the risk for most drugs. So let me change gears one more time and talk about the role of genetics in drug efficacy. So I'm going to tell this story, let me take you back to GSK's experience. GSK has been conducting pharmacogenetics research within our clinical trials and even post-clinical trials for marketed medicines since the days of Abacavir hypersensitivity reaction. We've carried out several dozen pharmacogenetic studies of various drugs for both drug safety and drug efficacy. And here's what we've discovered over the years. We have the Abacavir discovery, tranilast, and the effect of UGT1A1 on bilirubin. We found that a genetic factor affecting bilirubin in pazopanib, drug-induced liver injury for lapatinib, and drug-induced liver injury for pazopanib. All of these 5 discoveries that we've had within GSK to date are safety related. As we saw these patterns emerge, we asked ourselves, does genetics really have a role to play in safety pharmacogenetics? And is that role important enough that we would incorporate that into our clinical development activities? So to ask that question, we both looked quite carefully at the work that we had done previously, but primarily we looked outside. We asked how many examples are there out there of efficacy effects, genetic efficacy effects on drugs. What can we learn from those examples, and how should we go about finding them? And these results were published just this past year in Nature Reviews Genetics, which I can refer to you to if you'd like to get more details about that work. Let me just summarize one of the key studies and outcomes from that. I'm waiting for that slide to come up. A little bit of a delay here. Here we are. Okay. So to carry out this study, we went back to that Jiwa's catalog that I told you about before. And the study that we published had data in the catalog as recent as July of 2015. So there were roughly 2,200 distinct genome-wide association study reports in the catalog. We filtered those 2,200 down to those that were related to drug efficacy, of which there were 76, so roughly 3.4% of the catalog entries. Those 76 GWAS corresponded to 41 different drugs and drug classes or combinations. And of those, we filtered it down to those that had very strong statistical evidence underlying that association. And so of those, we found 13 GWAS, genome-wide association studies with drug efficacy that had 1 or more high confidence associations. That is only about 15%, 6 out of 41, of the drugs for which there was a genome-wide association study for drug efficacy did we actually find a convincing association. So what underlies those reported associations? So they're shown here. They actually correspond to only 6 distinct drugs or drug classes: warfarin, clopidogrel, rosuvastatin, other statins, metformin, and PEG interferon. The efficacy measure that underlies that each of those is shown here in the second column, and the third column shows the genes that had been identified as the likely culprit underlying those genetic associations. I've asterisked the entries here where there is a CPIC treatment guidance available in the CPIC resource I mentioned before, and that corresponds to warfarin, clopidogrel, and PEG interferon. So of all of the 76 GWAS that were carried out, these are the only 13 associations that we were able to identify from those studies. And again, those only correspond to 6 distinct drugs or drug classes. Several of those associations had actually been identified in the pre-GWAS era through candidate gene studies. For example, the vitamin K receptor association that I had described before was actually discovered not through these genome-wide methods, but through candidate gene analysis. We expanded our search not just to those discovered through GWAS, but also to those that had been identified through these types of candidate gene studies, but were nonetheless fairly convincing in their evidence underlying them, and I'm showing these associations here. So you have codeine, tamoxifen, eculizumab, the tyrosine kinase inhibitors, platinum-based chemotherapies, olaparib, and sulfonylureas, and these are the genes that have been associated with those. So let me just take a couple of minutes to summarize what we found with on the efficacy genetics side. We found 3 different types of genes that tended to be involved. Genes involved in ADME, again, the drug absorption and metabolism and excretion. Genes involved in the function of the drug, so the drug target itself, such as the vitamin K receptor. And genes involved in disease mechanism, so the disease much more broadly, as illustrated here. Interestingly, of those discoveries, 74% of the discoveries came about when there was a continuous biomarker that measured the response. So something that was relatively sensitive and relatively objective in measuring how well a patient is doing when they're on the drug. So we found that as quite telling from a study design perspective and how we want to go about pursuing and identifying these in the future, that we're much more likely to be able to identify them when we have a measure that's very sensitive to response, as opposed to a clinical assessment of responder versus non-responder. In the interest of time, I'm going to skip this relatively complicated slide and go on to one of the key outcomes from the study that we published, and that was to get a sense of, well, how many drugs do we think are likely to have genetic factors that influence efficacy that could be large enough that they could impact clinical decision-making. And so we took essentially our estimate of what proportion of GWAS we're able to find a genetic effect underlying it, the proportion of those known genetic factors that are large enough that it's likely to have clinical impact, which is roughly 64%, and then came up with this estimate that roughly 10% or so of drugs are likely to have genetic factors that are large enough that they can be discovered and could potentially influence patient outcomes. So that's our expectation going into this, that only about 10% of drugs are likely to benefit from genetic information in making decisions about which patients should receive it from an efficacy perspective. I'm going to skip this also just out of the interest of time. So some of the lessons that we've drawn from our efficacy genetics assessment, that we need to recognize that when we're searching genome-wide, that by conducting a very large number of tests, we're going to adversely affect our statistical power to identify those genetic associations. So if we can reduce the number of tests we carry out to a smaller number, we will increase our power to find them. And given that the mechanisms underlying both safety and efficacy effects are limited to a relatively small number of genes in the genome, at least that we've discovered to date, then focusing primarily on those mechanisms and modalities are likely to improve our success rates. We find that there are very few situations where pharmacogenetics is large enough that you would actually find it in a clinical trial and that it would rescue a failed trial, a trial that didn't have a large enough drug effect to be able to differentiate the treatment arms in the trial. Trials with larger drug effects are going to have more power to find efficacy pharmacogenetics. So this is a bit counterintuitive, but related to the previous point, and that is that we're going to have the most success in finding genetic factors when drugs are really effective. Of course, if they're effective for everyone, then we don't necessarily need genetics, but that's not usually the case. Even very effective drugs often have large subgroups that don't respond particularly well. And if we're conducting this in a clinical trial setting, we really do need to combine data across trials to increase our statistical power to identify them. So just to recap, implications on drug development, we expect about 10% of drugs to have genetic predictors of drug efficacy that could influence clinical decision making. Right now, the relatively low cost of pharmacogenetic analysis does justify, in many instances, very early and more routine screening for genetic factors, even in clinical development in phase 2 and phase 3 clinical trials. And that there are a number of things, as I discussed in the previous slide, that we can do to improve our probability of success and tailoring our analysis strategies to identify those factors. So, I think that I've made all of the key points for this presentation in these previous slides. So these slides are going to be provided to the participants in this. So I'm going to skip over my last couple of slides. You can find essentially a short summary of our overall observations of the role of genetics in drug exposure, drug safety, and drug efficacy in this slide, and I will just finish by highlighting the many people that have contributed to the work that I've presented today, both for the Serious Adverse Events Consortium and drug safety, as well as my colleagues here within GSK and the work that we've done around drug efficacy. Thank you.
Thank you, Dr. Nelson, for that informative presentation. Now, before we get started on our Q&A session, I would like to remind our audience how to submit questions. You can submit the questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of your presentation window. We'll try to answer as many questions as we can, but if by chance we're unable to get to your question due to time constraints, Dr. Nelson will be answering questions via email following the presentation. Okay, Dr. Nelson, our first question is, how many significant genetic variants are found in a typical Jiwa? Is it different for studies of diseases versus drug response?
Yeah, that's a really good question. So the examples that I showed here today, there were very few. So for example, the flucloxacillin GWAS or the augmentin GWAS really found just one major peak of association around HLA. For most complex diseases, particularly when we are able to expand those out into very large samples of cases and controls, we typically find that there are dozens and sometimes hundreds of genetic loci that are influencing these various diseases that we study. I imagine that if we were able to carry out a drug response GWAS with tens of thousands of patients who had taken the drug, and we were able to measure very well how they were responding to those drugs, we would probably find many, many more genetic associations than what we're finding in the kinds of studies we can carry today, where we're limited to a relatively small number of patients. So I don't know if that means that the underlying genetic architecture for drug response is that different than disease, or if it's just that we don't yet have the sample sizes and the statistical power to be able to find many of the very small, subtle effects that might be having relatively small impacts on drug response. But that's a question that hopefully one day we will be able to answer, particularly as genetic data becomes more routine as part of a medical record, and we may have the opportunity to combine both routine genetic data with large electronic health records to be able to search for those on a very large scale.
Now, Dr. Nelson, the Serious Adverse Events Consortium looks like it has been pretty productive. Is it still running studies?
Yeah, so the Serious Adverse Event Consortium was initiated in 2007, initially with a 3-year phase 1. In 2010, we decided to extend the consortium for an additional 3 years through very careful and use of its funding. The consortium has continued to function. Right now, we are in essentially the sunset years of the consortium. There are no new funds coming in for consortium research, but we do have sufficient funds to wrap up the research that we started. The main body of work going on right now is finishing up some extensive work around liver injury, around renal injury, as well as conducting a bit more research looking across different types of serious adverse events, as well as across drugs. But that work will be wrapping up likely within the next 6 to 12 months.
Now, Dr. Nelson, in your presentation, you briefly mentioned that pharmacogenetics is unlikely to rescue a failed clinical trial. Can you clarify what you meant by that?
Yes. So, and I'm there specifically referring to trials that fail because the drug just didn't have enough efficacy. We weren't able to differentiate the effect of the drug versus whatever the standard of care was, whether it's placebo or some other treatment. So the idea behind that is that if, excuse me, I'm drink of water. Sorry. The idea behind that is if there's not a large enough average effect, there most likely won't be enough patients within that trial experiencing a sufficient effect that even if there was a genetic variant or some other biomarker that could differentiate those patients from the rest, that you would actually be able to discover that in a clinical trial. That's coming back to that paradox that I mentioned earlier when I was introducing that, and that is that we actually have the greatest capacity to find those factors that differentiate patients that respond really well versus not so well when a drug is really effective. And when a drug is ineffective, there's just a very small number of situations where you could, particularly in an exploratory setting where you don't know the factors that may be differentiating drug response, to actually go in and explore and search and find them. So that's essentially what I meant by that.
Okay, thank you for that clarification. Now we are almost out of time. We're going to have time for one last question, but again, to our audience, Dr. Nelson will be replying to questions via email post the presentation. Our final question, Dr. Nelson, do you expect genetics to play an important role in the future of healthcare?
Yeah, that's the million-dollar question. My sense is the answer is yes. I think that there are enough examples, and again, referring back to the CPIC, the pharmacogenetics guidance that's available. There are enough positive examples to tell us that yes, genetics does have a role. The challenge is really in the economics of it. At what point does genetic information become just a routine part of a person's medical record? Because it's a very different decision to make when you are going to make a prescribing decision and you can see what drugs are likely to be most effective or safest in that particular patient immediately, as opposed to having to go out and conduct a laboratory test, which sometimes can be time-consuming. They can take several days or even weeks to get a result back, and they can sometimes be expensive, tens to hundreds of dollars for those tests. So my sense is that once genetics becomes a routine part of our medical care, that pharmacogenetics will find its place in routine medical care.
Dr. Nelson, I want to thank you today for your presentation. Would you like to say any final comments to our audience before we close?
No, thank you. I do appreciate the opportunity to present today.
Well, we appreciate it, and we appreciate you coming and taking your time out. I would also like to thank our sponsor, LabRoots, for making today's educational webcast possible, And also a thank you to our audience for their great participation. Today's webcast will be available for on-demand viewing through May 22nd, 2017. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. We thank you for joining us and hope to see you next time. Goodbye.